Well, all right, this is the last video of chapter 4. We're talking about on this DNA transcription and translation, basically how the body makes proteins. So protein synthesis, again, remember protein synthesis, two big things to remember before we go on. DNA, a gene, is the recipe to make proteins, and in the cytoplasm we have these things called ribosomes, which are the site of protein synthesis, or making it. So those are pretty important to remember. So the genetic code is a sequence of base pairs in a gene that specifies the order of amino acids to make a particular protein. Each amino acid is represented by a specific sequence of three nucleotides on the DNA. So we'll see that in just a second. The genetic code and the nucleotide sequences also represent stop and start messages. So we're going to have all of these things. Remember, the base pairs were adenine and thymine. They always went together. Cytosine and guanine, they always went together. As they form these little chains, these little things that connect along the way, each group of three represents an amino acid. And they also in there have special three group things that denote a start and a stop. Now, the process of protein synthesis requires two steps. One is called transcription. The word literally means to write down. So if anybody's taking notes and you're writing down these little things, that is transcribing. You have gone through transcription. Now, if I want to make whatever I wrote down something that is useful, and it's not useful to start with, I have to translate it. That's why the second step is called translation. So I'm going to copy down the recipe, and then I'm going to take the recipe and turn it into a product. I can copy down a recipe to make buttermilk biscuits, but it takes flour and buttermilk and to get in there, and butter or lard or whatever to get in there to make the biscuit. Writing it down is one thing, making it is another. Writing it down, transcription, making it is called translation. DNA molecules cannot leave the nucleus, but protein synthesis happens in the cytoplasm. So we're going to have some go-between, and the go-between is called RNA. It is shorter, obviously much shorter than DNA. It is going to be the length of the gene, that specific section for the protein. And so RNA molecules, as it said, differ from DNA molecules in a couple of ways. One, the main one, is they are single-stranded. Right? They're not going to have the two sides that kind of bind together, back and forth. RNA is single-stranded, and you're going to see it'll have the little bases on it on one side. So it is going to have ribose instead of deoxyribose, so that's why it's called RNA instead of DNA, deoxyribonucleic acid, DNA, ribonucleic acid, RNA. And the other thing, and the big one for us, is instead of thymine, it's going to use uracil. All right? So on the RNA, there is no thymine. Uracil is what is going to be on there. Now, this one that's going to come out is called the messenger RNA. This is the one that's going to do the transcribing or writing down. And so we're going to make this in the nucleus, and it will find its way in between the gene, that specific part. The gene's going to open up. This is going to work its way in the middle of it, and then copy down the base pairs. Except anywhere there would be a thymine, it's going to use a uracil instead. Now, wherever there's an adenine, it'll still be there. The only difference is no thymine, it'll use uracil. And again, this process is called transcription, making a copy. Now, RNA polymerase, which we looked at DNA polymerase in the DNA replication, RNA polymerase helps direct the synthesis of messenger RNA. Now, as we're going through and doing this, RNA polymerase is going to recognize the beginning and the end of the gene, and so that helps open it up. So as we look at this, there's going to be, this is kind of showing that I'm not worried about this right now. I mean, this is the same thing we talked about. It's got uracil instead of thymine. It is single-stranded instead of double-stranded. It is ribo instead of deoxyribo. So this is just kind of going through stuff. These are the complementary pairs. I don't like the way this puts it. What it's saying is, if I have on DNA, if I have this here, and it makes sense, wherever there's an A, there's a T. Wherever there's a T, there's an A. Wherever there's a C, there's a G. Wherever there's a G, there's a C. Right? That's how the DNA works. Now, if this is one strand of DNA, right? Now, on the other strand of the DNA, if I was to go over here and say, if I'm looking at the other strand of DNA, it is an A and a T. Whoop, that's supposed to be a T. This is a T and an A. This is a C and a G. So this again is the DNA. Now as the RNA polymerase comes in, it's going to open this. It's going to slip in, the messenger RNA slips in, and it makes a copy. Now, since there would have been a T, as you can see on this side, since there would have been a T here, and on the messenger RNA, you cannot have a T. We have a U for uracil. Now, where the T is on DNA, it's the same as an A. If the C is, it's the same, it's a G. Where the G is, it's the same, it's a C. The only difference is wherever there would be a T on the DNA, there's a U, all right? On the other strand of DNA, I should say, on the complementary strand. Now, translation is going to happen outside of it. Translation means I'm making it useful. So if I gave you the test a week early, but I wrote it in like Mandarin Chinese, since most people probably do not speak Mandarin Chinese, you'd have to go home and find a translator. You have to turn it into something useful, and that's what's happening here. These little base pairs, adenine, thymine, or adenine, uracil, cytosine, and guanine, these, even though they represent amino acids, they're not amino acids. So each one of these is a set of three called a codon. Now, we're going to have a start and a stop codon. I'm not worried about that. I hope you know that the start codon means it's the start and the stop means the stop. But once the messenger RNA has made this copy, this transcription, it will leave the nucleus through a nuclear pore, and it'll find the first free ribosome. Now, where it gets the ribosome, it's going to start being pulled in three little sections, which we call a codon, at a time. And it's going to be read. All right? It's going to be read by something called transfer RNA. This is a process called translation. The transfer RNA is going to look at the codon, and transfer RNA is much smaller, by the way, but it's going to look at the codon. It's going to make something called an anticodon, and it knows what amino acid is represented by this anticodon and goes and gets it. It's meant to basically say it transfers the amino acid into place. So translation, the ribosome holds the mRNA in position, the messenger RNA in position. And so it's almost like I've got this ribosome with this little skylight. There's two little parts to it, but as it's pulling this in, each codon is held in place underneath this skylight. And then the transfer RNA comes in, reads it, makes an anticodon, goes and gets the amino acid, right? And then comes back again and puts it in place. And we're going to see a picture which is much better than mine in just a minute. Now, there are 20 types of amino acids. So imagine that I've got a box full of Legos, and there are 20 different colors. Each anticodon represents a color. So I look in, it's the transfer RNA says it's yellow, so it makes an anticodon, gets the yellow Lego, brings it back. The next one comes in, it looks at that one, and it says, this one's blue. It goes and gets the blue amino acid, brings it back. And that's how it works. Now, as these amino acids are joined together, so peptide bonds, a new protein molecule starts being created, and it starts folding into its unique shape, and we call that conformation. Now, again, these are kind of showing the different sequences. This is another thing I am not worried about you knowing, all right? This picture is probably the best picture kind of showing you how things are happening. This over here, this again is transcription. I'm writing it down. Messenger RNA, you see right here, weaseling into this DNA, and this would actually be this one section here to here. This would be considered basically the genome, or the gene, not the genome, the gene that it's trying to read. Now, as the messenger RNA gets in there and makes a copy of it, all right, again, as it's making a copy, it's going to be the identical base pairs as on the opposite side, except for the uracil. Wherever there would be a T over here, there will be a U over here. Now, as it makes it, it's going to finish up. It will leave through a nuclear pore and find a ribosome. As it's going through, the ribosome is going to look at the groups of 3, which we call codons. It makes an anticodon and then goes and gets the amino acid that's supposed to be there and brings it back. You see them doing it here, one after another, and then we have a peptide chain being formed. Now, as this is going on, again, this is looking at it one after another. It's pulling it through. These are showing all the different codons as it's going through and being read, translated, and turned into a protein. Now, in proteins, remember, they have a very unique shape, and so they have to fold just right. Now, there are things that help make sure our body, that these proteins are folded in the correct position. Any little bit, they're not good. Misfolded proteins, as it says here, are either refolded. We'll have, again, a little enzyme that goes through and tries to refold it. If it's not, it's going to be destroyed. You don't have to know proteasomes. These are the ones that go through and they just break it down. A gene that has been transcribed and translated into a protein is said to have been expressed. We have expressed a protein. I've made it. Those two parts, transcribed, which happens in the nucleus, and translated, which happens on the ribosome, together are what we describe as a protein being expressed. And so again, gene expression is based on cell differentiation. All this is saying is I'm going to have certain cells that are in the kidney. Right? I'm going to have certain cells that are in my eyeball. I'm going to have certain cells that are in my fingernails, whatever, all over my body. And they have the exact same DNA in it, but the gene expression for each cell tissue is going to be controlled by cell differentiation, the different needs. And that's all it's saying. The main thing on this is I want you to know transcription and translation are how genes are expressed or made. Transcription happens in the DNA, or in the nucleus of the DNA. It is involving something called a messenger RNA that makes a copy. That copy leaves the nucleus through a nuclear pore, finds a ribosome, and starts being pulled through. Those 3 individual bases that we call codons, as they're grouped into, as it is looked at by a transfer RNA, an anticodon is made, and then it goes and gets it and puts it back into its spot. I hope that makes sense, and I hope everyone has a great day.